hydroxyl there. And that again applies to these exonuclease questions, because the only important part for an exonuclease question is where the phosphate is. Okay. All right. Let's see. Where were we in the PowerPoint? Exonucleases, restriction enzymes. All right. Restriction enzymes. General information is needed for exam 3. All that stuff about recombinase, cloning, and everything that I prepared you for won't be on exam 3. So, I will get to it again in a second. Don't worry. Basically, first, to introduce you to the concept, restriction enzymes. You should know the general information of a restriction enzyme. A synonym for restriction enzyme is endonuclease. So what a restriction enzyme does is there's a particular pattern. Let me find a nice example, I guess. Visually, this is to illustrate what a restriction enzyme will do. Here's a restriction enzyme called EcoR1. What it does is it looks all across a particular genome given for this specific pattern drawn on here. You don't have to memorize what the pattern is, just know that a restriction enzyme looks for a pattern in the genome, and every time it finds that pattern, it will perform a cut in DNA. And the end result usually is DNA will open up, or a fragment of DNA will be cut out of the genome target. So that is what a restriction enzyme does, is it looks for a particular pattern and it cuts it. That is what a synonym for restriction enzyme is, endonuclease, which is what we mentioned earlier. Other general information that's important is, where is it? Here is the general information of like type 2 versus type 3 and type 1 restriction enzymes. It's kind of given to you right here, so I'm not going to go over it in detail, but be able to, in a multiple choice version, be able to tell the difference between them. Really, the important part is this thing here, is a type 1 restriction enzyme is unable, it does not have a specific pattern it looks for, all the other ones do. Let's see. Also, other differences. These are listed on here for you. I'm not going to go over it in detail, but look at the differences between them, and only the information that is given to you on the PowerPoints will be asked about in regards to restriction enzymes. So don't worry if there's something missing here, you won't be asked about it. Yes?
So can it also cut when the sequence is reversed?
Yes. So that usually happens if it's like on the opposite strand. That happens all the time. Yeah. In fact, restriction enzymes require both strands of DNA to match the pattern that it's looking for. Then it will cut it. Let's see. This stuff. Yes. This may be asked about. So, this experiment is an experiment conducted called restriction fragment length polymorphism. Basically, the way this works is if you have two DNA samples, you can compare them to each other by what restriction enzyme sites they have. Basically, most people, actually everybody, only differs from each other by a few hundreds of bases out of the millions and billions of bases that are in your genome. So those subtle differences can be identified at the molecular level by where restriction enzymes cut at. In this illustration here, we have two different versions of this chromosome. This chromosome up here is expressing the dominant gene, and this chromosome down here is expressing the recessive gene. In genetics, we describe dominant genes with capital letters and recessive stuff with lowercase letters. And I keep saying genes, but it's alleles, whatever. This isn't a genetics class. But that's what these notations mean. This is capital A is dominant, lowercase a is recessive. Each of these arrows represents a site at which a restriction enzyme will cut at. They're obviously not going to tell you the pattern or anything because that's not the focus of the question. They're just telling you here is where some restriction enzyme will cut. That's also there and there. And on the recessive gene, on the recessive allele, it will cut there and there. Visually, you can visualize these cuts by the addition of some type of fluorescent probe, which on this diagram is illustrated by this black bar here, and then running it on a gel. You remember back in exam 1 when we talked about proteins and how you can separate proteins based off of the length or size of the protein. Well, over here, you can separate DNA fragments from each other by the length of the DNA fragment. So we can visualize that. We'll do this example over here. And it may be a different genotype than what's illustrated on here, but it's good to start with this if you're learning it. Is that if somebody only has the dominant alleles, if they only have a if they're only homozygous for the dominant allele, they should only have the capital A allele. What this means is it will only cut at the indicated sites and you ignore the recessive stuff. How will you visualize that on a gel? Well, if it only has this allele, it should only show up as one band because it doesn't have the other allele. So notice there's only one band in the gel there. The other fact is the fact that this fragment of DNA is short, right? Shorter fragments of DNA will be closer to the bottom of a gel. If you don't remember that fact from exam 1, I'm reiterating it. Smaller stuff, shorter stuff will be closer to the bottom end of a gel. Longer stuff will be closer to the top end of a gel. Okay? Yes?
So do you need like the fluorescent probe for this to work?
Yes, so this really small fragment, you actually won't see it on the gel because there is no probe attached to it. So make sure you look at where the probe is attached to help interpret this diagram. So, for someone who has capital A, they'll have one band and it'll be closer to the bottom. For someone who has only the recessive allele, they will have 2 copies of this recessive allele. They should show a band that represents the longer fragment of DNA. Notice that it is closer to the top of the gel. Someone who is heterozygous will have both of these things, so they should have 2 bands, one representing the shorter fragment and one representing the longer fragment. So be able to identify, if you're given this gel, which one is dominant, which one's recessive, or which one's heterozygous. You may be given this diagram on the left and the gel to help you identify it. You very likely won't be given the column names, so you will have to identify which column is homozygous dominant, which one is homozygous recessive, and which one's heterozygous. Okay? Other questions regarding this? All right. So, oh, yes. Mm-hmm.
Like, let's say if it's the heterozygous,
Yes.
could it also be like on the left, or it's always going to be on the right, as it appears?
You will have to see where the probe is, if
Okay,
that's
so
what you're,
basically-
or you talk about on the gel. On the gel or over here on the left?
gel, yeah,
On the gel now, they can mix up these
They could mix
lanes.
it up.
Yeah, you go by number of bands and where they're at in
Okay,
the gel.
okay.
Okay. Oops.
Do we have to know about the variable tandem repeats?
That's sort of similar to this. Don't worry about the specific details, but that's kind of why restriction fragment length polymorphism works, is that we have variability in our DNA based on where these there's a term called single nucleotide polymorphisms, is why we have these variability in DNA at where restriction enzymes cut. But don't worry about whatever tandem repeats. Don't worry about that. This will probably show, if historically speaking, if this doesn't show up on exam 3, and it probably won't, it will show up on the final. So we will review it in more detail later.
Is that what you went over as far as like the gene? Or
Yeah, and with like the A cut, B cut, or A site, B site, C site, all that stuff. We'll go over it again in more detail for the final. It is probably too much for exam 3 right now, okay? Which I'm sure is a great relief to a lot of you. So, skip. Sanger method. All right. You may be relieved to know that most likely the only thing you'll be asked about with the Sanger method is you'll be given a gel and you have to figure out what the original DNA sequence was. Don't worry about the mechanisms like the deoxynucleotides or whatever that used to be asked about. For now, the important thing is if you're given a gel to be able to sequence it. So let me make up something real quick as a nice practice. Let me draw something first and I'll flip on the projector.
Do we have to know about the lysogenic and the live cycle?
The bacteriophages, not for exam 3, no. Okay. So you may be given a gel representing the Sanger method, which is to sequence an unknown strand of DNA. The way this works is that the gel has several bands on it, each representing a particular base on the complementary strand of DNA. So basically, if you want to find the original strand of DNA, write out the complementary strand of DNA, and then base pair it to figure out the original strand of DNA. So the question will be, what's the original? And this will only give you the complementary. So there are several ways of solving this. I personally want to build the complementary strand of DNA from front to back. Okay? There are other ways of doing this, but that's how I personally do this. The very front of any newly synthesized strand of DNA must first contain the primer. So the primer is always on your new strand of DNA. It's what starts everything. Okay? The primer will be given to you, and you would start out by drawing that. The rest of your new strand of DNA will be whatever the gel tells you. The question is, which end of this gel do you need to read? Well, remember that the bottom of the gel represents shorter fragments of DNA. What this means in terms of the Sanger method is that the bottom of the gel represents the first few bases in your new strand of DNA. The top of the gel represents the last few bases in your new strand of DNA. So, reading from bottom to top, and the columns will be labeled for you, the very first base that the gel prints out is another A. This one here is C. The next one from the bottom, next two are G's. That's an A there. That's a T there. And at the very top is another A there. This is your complementary strand of DNA. It is not the final answer. They want the original strand of DNA. So what you have to do now is you have to base pair this thing. All right, which is the easy part. T's. Because you know your base pairing rules, you know your char-gaps rules, very easy to get your original strand of DNA. And then there's one more trick. There will probably be one answer choice that looks like this. And there may be another answer choice that looks like this. Which one is right? B. Why is that? Answer choice B is correct because notice I have not labeled which end is 5' or which end is 3' on my answer choices. And you remember now that if nothing is labeled, you assume that the left side is 5' and the right side is 3'. Notice our original DNA sequence. This is just my personal way of doing this problem. The kind of downside to it is that I end up writing the original DNA sequence in the 3' to 5' direction. But because I remember directions, I can just flip this around and match it to the correct answer choice. Okay? Yes? Question?
I don't know if somebody asked this, but are they going to ask about the DDG, the DG?
So the actual mechanism won't be asked about on exam 3. So this is just a straight up figuring. This is the Sanger method, is just reading a gel and figure out the original DNA sequence. Yeah. Okay. Other questions? All right, pretty straightforward. Okay. There's a lot to skip.
We don't have to know about the bisulfite.
Nope.
Questions?
Yes.
Do we have to know how to be able to calculate how many cycles of DNA, how many DNA we have after cycles of PCR?
No, you just need to know PCR as a mechanism. Did I skip PCR? I just want to make sure. I think it's, if I skipped it, let me talk about PCR real quick. You won't have to calculate anything with it.
Bisulfite.
Oh, is that what the bisulfite stuff
No,
was?
it's before.
Oh, it's before it? Oh, I must have skipped it. My bad. Let's see.
Or no, it's after the, yeah.
Is this the PCR?
No.
No.
You skipped that.
Well, alright, wherever it is, it's very simple. It's pretty easy. Let me just draw it. So, PCR is a very important mechanism because it's used all the time in the lab. It's used all the time in biology because the goal of PCR is to amplify a nucleotide sequence. Okay, is to amplify a nucleotide sequence. The way it works is you have you have one copy of your nucleotide sequence, of your double-stranded DNA, and you can even do this with mRNA. We have one copy of it. PCR is 3 steps repeated over and over and over again. Step 1 is something called denaturation. Basically, we heat up the machine will heat up the sample, and what will happen is this double-stranded DNA will denature into individual single-stranded DNA strands, because at a high enough temperature you will break the hydrogen bonds keeping your DNA sequence together. The second step of PCR is called annealing. So it's going to mimic DNA replication in a way. The annealing process is, now that your strands of DNA are separated from each other, the annealing process is the attachment of primers to your separated template strands. And remember, the primers are always on the 5' end of your new DNA that's going to be made, right? So in fact, going by that, at one end of the template will primers be attached to? Well, if they're on the 5' end of new DNA, primers will always go to the 3' end of your template. So be careful with that. So the annealing step is the attachment of primers. The last step is elongation. Or maybe sometimes it's amplification. But basically, conceptually, what this really means is that, well, now that you have your primers in place, replicate your DNA. There's an enzyme that will conduct this called Taq polymerase. It's a special DNA polymerase that works at high temperatures. So that's why this only works in PCR, is because you don't really see this in many actual organisms. So it uses a special DNA polymerase called Taq polymerase, and the elongation step is you replicate your 2 strands of DNA. All right, and you started with 1 copy of DNA, now you have 2. And every time you do these 3 steps, it will double the amount of DNA that you have, so it increases exponentially. All right, and again, the purpose of this is to amplify a particular nucleotide sequence so you can do whatever experiments you want with it. Usually they do this to measure gene expression, but we won't go over that in detail. It's not necessary for the exam. So this is PCR. These are the 3 steps of PCR. Denature, to separate. Anneal, to attach the primers. Elongate, which means replicate your DNA. And this all occurs at relatively high temperatures. Okay? So that's PCR. Okay. All right. Before I move on to 11, let me check some stuff. All right, moving on to chapter 11. All right. All right, some important information on chapter 11. So, chapter 11 is actually a fairly short chapter that really describes some properties of DNA structures such as chromosomes and DNA as a whole. We're involving phrases like supercoiling. So, to start with, a property of DNA. And it turns out you can compare different samples of DNA to each other by their melting points. What I mean by that is, remember that DNA contains nitrogenous bases. Your nitrogenous bases are really good at absorbing UV light. The problem is that when DNA is in a double helix form, those nitrogenous bases are hidden on the interior portion of DNA, meaning they don't actually absorb UV light as long as DNA is in its double helix form. However, if you heat samples of DNA with temperature, if you increase the temperature, at some point your DNA will begin to unwind. That is the melting temperature of that particular DNA sample. If a DNA sample contains lots of A's and T's and very few G's and C's, it will melt a lot earlier. It will melt at a lesser melting temperature. And how will you visualize that? Well, on this graph, they call that hyperchromic shift, by the way. On this graph, all of a sudden, your DNA will be able to absorb a tremendous amount of UV light when it wasn't able to before. And that's because your double-stranded DNA has unwound into single-stranded DNA. And all your nitrogenous bases that were formerly hidden on the inside are now exposed to the environment, and they can absorb tons and tons of UV light. And that's indicated by this sharp increase here. Notice, in contrast, a DNA sample that has lots of G's and C's, which is this one over here on the right, you need to add way more energy, you need to increase the temperature way more before it is able to absorb UV light. And that's because it's harder to break DNA that has a lot of G's and C's in it. So that's one property that DNA samples have and how you can measure it. Another two kind of characteristics that DNA samples have is one, the rate of renaturation. Basically, the rate of renaturation is if you've separated your two strands of DNA and you decrease the temperature again, how quickly do they recombine? It turns out that if they recombine very quickly, that means they're very simple strands of DNA. And that's because what I mean by simple is very long patterns of just A's, or like a bunch of A's in a row, or a bunch of G's in a row, or a bunch of C's in a row. There isn't like a complex pattern of bases. So if renaturation happens quickly, it is a very simple strand of DNA. And if it takes a while to renature, it is complex.
I have a question.
Yes?
So renaturation wouldn't be affected by the GC pairing as opposed to AT?
As opposed to just complexity. Renaturation is more affected by just how complex the base pattern is, not necessarily how many G's or C's or A's or T's are. The number of those things only affects how easy it is to open the DNA, not necessarily to put it back together. Okay, so that's one feature of DNA is rate of renaturation. The other feature that you can analyze DNA by is homology, which is how similar two strands of DNA are to each other. If you have two strands of DNA from different organisms, you can track how homologous they are, how similar to they are, by seeing how well they hybridize to each other, how well they simply base pair to each other. We do these homology experiments to track evolutionary, to make evolutionary trees. When we're comparing organisms to each other, we see how well their DNA strands just simply bind together. If they don't bind very well together, if they poorly hybridize, they are very different organisms at the genetic level. If they bind, if they have a good hybridization, they are usually very similar in terms of evolutionary information. Supercoiling, okay. So supercoiling is a general consequence of DNA that happens whenever you open up DNA. You usually open up DNA via DNA replication or transcription, and every time you open up DNA, it builds up torsional strain everywhere else in that strand of DNA, in that sample of DNA. That torsional strain that happens is called supercoiling. Now, most of the time, we're able to fix supercoiling via two categories of enzymes called topoisomerases or gyrases. Technically, gyrase is a type of topoisomerase, but you can pretend they're two different things when you're studying for this. So topoisomerases and gyrases are special enzymes that fix supercoiling. There's a lot of information on these. Make sure you do know the different types of topoisomerases, the differences between them, and what gyrase does. Yes?
So when you actually unwind the DNA with helicase, there's no supercoiling created?
Yeah, when you unwind DNA with helicase, ahead of that area, there's going to be some supercoiling that develops. Because whenever you disturb DNA in any way, you create supercoiling elsewhere.
Okay.
Yes?
So well, normally what it does is that it unwinds the negative supercoiling by adding positive?
Yeah, so you have to look at the definition for it, but yes, topoisomerases in general, not every time,
Okay.
but in general work by adding positive supercoiling.
Okay.
And gyrases work by adding negative supercoiling. So it gives a hint at what types of supercoiling they fix because they add the opposite type.
So it removes the negative supercoiling.
In general, yes, which is all you need to know is the general information. Yeah.
So if helicase doesn't or creates supercoiling, but it doesn't alter the linking number, how does it do that?
Linking number is the current. How do I describe? Linking number is the current state that DNA is in. You never have just helicase and no topoisomerases or anything. So I know what you're talking about, but you won't be asked anything about calculating linking number for something undergoing DNA replication. The linking number stuff is like how do I explain? You basically have to answer that in terms of its own separate concept. So don't worry about what helicase affects linking number or not. You have the information here, but they won't ask you to say, you use helicase, what happens to the exact calculation with linking number? You won't have to worry about that. Just general features like this. The only time you have to calculate anything is when you're given some information about twists and writhe, and if they use the phrase relaxed or not. Okay, so I'll explain this. This formula here is a way to quantify the strain, the supercoiling, that a particular strain of DNA is experiencing. Basically, there's a number called linking number, and it is the addition of the number of twists that DNA has, and the writhe, which is kind of hard to illustrate, but basically writhe is when you take your, if you have a rubber band and you just turn it around, the number of times you've turned around that rubber band is what writhe is. So imagine that DNA can do that too, that prokaryotic DNA can do that too. You can sort of see that illustration right here. This represents the writhe of a sample. If you're given a picture of DNA, you won't be asked to calculate how much writhe there is in there, so don't worry about trying to figure out how much writhe there is in something. Yes?
So for the other example that you had, so if the original number is 40 and then you made L 44, does that mean W is 0 or W is negative or W is positive?
In that case, twists don't change, so yes, writhe will have to change.
So twists never change?
Right. When we do these calculations with linking number twists, there's probably some example somewhere where it does, but for the purposes of undergraduate biochemistry, we don't change the number of twists in something. Okay, it's just the writhe that changes. So how to solve this type of thing? Basically, you have a sample of DNA that is some base pairs long. It is B DNA, and even if they don't tell you it's B DNA, you assume it's B DNA, which what that means is there's always going to be 10 base pairs per turn, 10 base pairs per twist. So if something is 400 base pairs long, and it is in the relaxed form, you can calculate the number of twists and the number of writhe just from that piece of information. Whenever you see the phrase relaxed, immediately write down W equals 0, okay? Because that is what relaxed DNA is. It has no writhe in it. You can calculate the number of twists based off how many base pairs the DNA has and divide it by 10. That will tell you how many twists it has because that's what bDNA is. It's 10 base pairs per twist. Now that you have L and you have T, you can calculate the linking number. If there is any supercoiling added, you will have to investigate your linking number, your twists, and your writhe to figure out what type of supercoiling is currently being experienced by that DNA. If there's negative writhe, what happens to linking number? It's probably going to become negative. Not negative. It's going to be lower than what you'd expect. If you have positive writhe, it's going to be higher than what you expect. If you have a greater linking number than what would be calculated like this, with 0 writhe and whatever amount of twists. You have a higher linking number than expected, that's describing positive supercoiling. And if you have a linking number that's less than expected, that is negative supercoiling. All right, so enough with supercoiling. We don't need to go more in detail about supercoiling. The next concept is cruciforms. You may be given, and we practiced it at this at SI, you may be given several different DNA sequences, and you may be asked which one will form a cruciform. And in some cases, you may be asked which one will form a strong cruciform, or which one will form a weak cruciform. So first, you have to figure out if the DNA sequence you were looking at will form a cruciform in the first place. It's very simple. You look for what is called a palindromic sequence. And, let me zoom in here. Okay. A palindromic sequence is simply, if you read a sequence from left to right, and you go to the opposite end, and you read that end from right to left, if they are the same pattern in those two opposite directions, that is a palindromic sequence. So see in this example, GAA, CG, TCC. Over here on the exact opposite end, you see GAA, CG, TCC. That is a palindromic sequence, and that means this region of DNA is very likely to form a cruciform, which is this cross shape that you see over here. Basically, what a cruciform is when a DNA strand accidentally binds to itself rather than the complementary DNA strand. So you see that illustrated here. And this only happens with palindromic sequences, yes?
So is there a difference between a palindrome and an inverted repeat?
It's synonyms of each other. Palindromes and inverted repeats are synonyms of each
So
other.
when mRNA binds to itself, is that like a palindrome or?
It is from a palindromic sequence also. You're talking about a hairpin loop,
Yeah.
right? Yes, that is also from a palindromic sequence,
Okay, so
exactly.
palindromes can form either cruciforms or...
Or if we're talking about mRNA hairpin loops, yes. And I'll talk about hairpin loops again when we get to transcription. All right. So, eukaryotic DNA has this extra component called nucleosomes, which are nicely illustrated here. So in eukaryotes, when we have DNA, they are compact. They are stored in a compact structure called a chromosome or chromatin. What chromatin is, or what chromosomes are made of, are a bunch of DNAs, or is DNA wrapped around these proteins called histones. That unit of DNA wrapped around a histone is called a nucleosome. And you can modify nucleosomes. The body actually naturally has different modifications to nucleosomes. Some regions in nucleosomes have methyl groups. Some regions in nucleosomes have acetyl groups. Some regions in nucleosomes are low in methylation, and some are low in acetylation. So there's a bunch of different ways you can modify nucleosomes in eukaryotes. These modifications generally are linked to changes in the expression of genes. If you have a gene that's wrapped around a histone, and that histone is very methylated, would you say that gene is highly or low expression? Low. Methylation, methylated DNA is associated, and I am oversimplifying it, but for this class it's enough. Methylated DNA is associated with low gene expression, yes.
So if the DNA is wrapped around the histone, that itself will be there, right?
The reason why I state that is that even when DNA is wrapped around a histone, we have transcription factors that help unwind it. So I don't want to go into detail too much.
Eukaryotes only, right?
Yes, and the reason why I don't want to go into details, that's 29B more goes into detail with
ask
that.
a question like, oh, if this is wound, because isn't that like the... I don't know.
He will ask general stuff like this in terms of like methylation versus
But
acetylation
not
and
the
how.
itself being around the histone.
Right, it's just the methylation versus acetylation stuff is the more important stuff. So again, what the important takeaway here is that if you have DNA or a region of DNA or a gene that is very methylated, it is usually not very highly expressed. And if something is acetylated, that's the opposite. If something is acetylated, it is usually more highly expressed than usual. Yes?
So what about the ESBAF? Because that's basically making it around the histone or not around
Yep.
the histone.
So,
So that turns off transcription or on.
it is not illustrated on here, but this thing affects the pattern of methylation and acetylation on here. So, next feature to talk about is this complicated looking thing, but I'm going to simplify it, called a ESBAF or BAF blocker. Basically, this enzyme's job is to modify nucleosomes in your genome to help the alteration of gene expression. What this thing normally does is it shifts nucleosomes around using some complicated methylation, acetylation stuff that you don't have to know about. It shifts nucleosomes around in order to alter what regions of the genome are open and what regions of the genome are closed. In this example here, if it's working normally, it shifts the nucleosomes around so that an enhancer region is open. And I know we go over enhancers in more detail in 29b, but because this particular information is found in chapter 11, it is fair to ask about on exam 3. So, don't worry about what an enhancer is. Know that when this is exposed, the expression of this particular gene is allowed to happen. And again, it happens because the nucleosomes have been shifted in a way that the enhancer is now open, and you can express whatever gene this is matched to. Okay? What a BAF blocker does is that it inhibits this ESBAF protein, so it can't shift around the nucleosomes. If it is unable to shift around the nucleosomes, this enhancer, look at that, is still wrapped around the histone protein, shutting off the expression of whatever gene that it is matched to. Okay? And that's, described here and illustrated up here. In fact, they go into slightly more detail and say that if you use this BAF blocker, you don't create the correct mRNA. You create this useless non-coding RNA because the enhancer has been is partially closed now with these nucleosomes not being moved out of the way. Okay, so that's illustrated here. You may be given multiple choice questions that ask about this stuff. I'm going to move on though. All right, let's see. Philadelphia chromosome. Yes, question.
So for the fish and the leopard, so what's the purpose of that? Just to see if there's a mutation or?
So it won't be asked about in detail, but lab-wise, what it is, fluorescent in.
If it binds, then it's mutated, but is that?
It's a little bit more general than that. So, fluorescent in situ hybridization, which is called FISH. The way this works, the way it's applied in is to find mutations in DNA samples. But what the mechanism actually is there's a fluorescent probe with some nucleotide pattern,
If it's color, then.
and that probe will go and bind to whatever target sequence that it matches to. Usually we design these probes to look for mutated versions of genes, and so visually, we usually see that in some fancy colors indicating that this probe has found a mutated version of a gene. What is important for this, and because FISH is applied to this next concept, which is the more focus here, is something called a Philadelphia chromosome. So, the Philadelphia chromosome is not a real chromosome. It really only exists because one chromosome, I don't remember what number, you don't have to remember the number, one chromosome accidentally fuses with another. And it creates a gene that normally doesn't exist. It creates a gene that was made out of a fusion of two separate genes. And the result, and the important part, is the result is that you create something called an uncontrolled tyrosine kinase. So what tyrosine kinase normally does is it goes around phosphorylating a bunch of things. In general, that causes the activation of a bunch of proteins. And in fact, what an uncontrolled tyrosine kinase will do is it will go turn on all those genes necessary for cell replication, which is bad because if this is uncontrolled tyrosine kinase, it means you have uncontrolled cellular replication, which we know there's a term used to describe that, cancer, right? So this leads to some particular type of cancer. I can't remember what type. It might be listed on here. Don't worry about the specific cancer. What's that? It's some type of blood cancer. I don't remember the name of it, though. All right. Yes?
Is it important to know that it comes from fused chromosomes?
Yes, that the Philadelphia chromosome is not a real one. It is a fused one. It's a fusion of two separate chromosomes
So
together.
translocation also, right?
Translocation is close to that, and I know it says it on here, so might as well use that term for this case. Translocation is technically a more general term, but yes, this is a translocation.
Ask if it's fusion or translocation.
It won't be that specific. It'll be a fusion chromosome. All right. All right, let me check stuff.